Hi there. Hi. Good evening. Welcome to the Museum of Science. Welcome to our inaugural program for our fall 2015 season. It's exciting to be started back up, and it's nice to see you all here. Great crowd. Please do the usual now. Take a moment to turn off your cell phones and devices. If you need to leave before the program is over, please exit through the door up the stairs and out the rear of the theater. This helps to minimize interruptions. In honor of the museum's Hall of Human Life, Lee and Niall Albright have established a way for us to spotlight notable scientists researching human health and biology. We are honored to host this annual symposium and look forward to many enlightening evenings about cutting-edge research, one of which we are revisiting tonight. The microbiome is now known to be as significant to our health as are our genes. This population of trillions of microbes cohabiting our bodies may hold the key to maintaining good health, both mentally and physically. We're excited to welcome back to the museum two eminent scholars, both of whom have graced this stage before. Dr. Tim Spector has blazed a trail with his study of identical twins and diet, and he will give us the latest on what he's learned about how the foods we eat influence our health and our gut. Dr. Eric Alm advanced the field's knowledge of the microbiome by studying his own every day for a year. Nicely done. Now he's focused on how we can manipulate our gut bacteria for better health and actually turn our bugs into drugs. Each gentleman will present their work, and then they'll have a conversation together. Following that, the audience will have a chance to ask questions. And following that, if you're interested, Dr. Spector will be signing copies of his new book, Behind the Curtain. That's not the title, but it will take place behind the curtain. Now, we're thrilled to bring together these luminaries who are discovering new information about our gut microbiome. Before we dive in, Dr. Albright would like to say a few words. Please join me in giving a warm hand to Dr. Niall Albright.
Welcome, and what a great pleasure to be leading this symposium again, which is in honor of the Hall of Human Life. You all have an assignment, and that is to go in this coming year at least once through the Hall of Human Life, and I know you'll enjoy it because of the interactive exhibits. And this museum is a pro on interactive exhibits. There are over 700 of them, in addition to 200 other exhibits that go on at the Museum of Science, and that's why we say it truly is a world-class museum. This is a great privilege for us. Last night, my wife Lee and I had a delightful dinner with both Tim and Eric and learned the inside scoops. You're going to hear about them tonight, and it's a first time that this symposium has been across the ocean on both sides. Tim Spector is in London at Guy's and St. Thomas' Hospital and is a genetic epidemiologist that has put together what I think is so remarkable, over 12,000 pairs of twins, girls and boys that they study ongoing to learn what's different about one twin from the other. Why is one heavier? Why is one brighter or taller or does things differently? So those are important things to study. I'm pushing Tim to tell us why cancer develops in some people and not others, as I'm the president of the Advanced Medical Research Cancer Foundation, so it's of intense interest. But one other thing that I really am very proud to honor tonight, and that is my mentor and the founder of this museum, Bradford Washburn. And this really goes to the heart of things because he came to our house to wish my sister Tenley Albright off for the Olympic competition, and he noticed a little picture of me when I was 15 climbing in the Blue Hills. He turned around, and as he left, he invited me to join him, and soon I was 6 weeks in the same tent with him and Norman Reed, so we got to know each other well. at the foot of Mount McKinley and then onward to Mount McKinley, now Mount Denali. So the Washburn family we're extremely grateful to, and that's what I'd like to dedicate this symposium that we do every year, just to let others know how important the director, founder, and inspiration of this museum was. We have two keynote speakers tonight, which is the first time we've ever had more than one. Our first speaker spoke the first year, some 2 years ago here, and talked about how identically different we are, and that was one of his books that he wrote, Identically Different. And then if you ever pick it up, it says how we can change our genes. What a relief. We think we're born that way or born with cancer, but he has demonstrated elegantly through his work at King's College, Guy's Hospital, St. Louis, and managing 70 other colleagues in London. So you're going to be treated to something very special you won't find any other place. Our second speaker is so prominent in the Boston community that he is starting the new Center for the Microbiome at MIT. He also deals, we learned last night, with 500 other centers that are trying to learn in the United States about our bacteria, bugs, the good ones and the bad ones. So we expect great things for them. And I'll just close by turning this over to Tim Spector, welcoming from London to here.
Well, good evening, and it's a great pleasure to be back here again and talk about my favorite topics. And I'm the warm-up act before Eric gets on and gives you the detail, and I'll do my best to give a nice, gentle overview of the problem. And this, as I see it, is the problem. This is the fact that nowadays we're obsessed with what we eat more than anything. More and more diets are coming out every week. And despite this obsession with our food and our diets, in the last 30 years, rates of obesity have tripled. And at the same time, doctors like myself have been telling my patients, it's simple. You just eat less, exercise more, calories in equals calories out, you'll lose weight. And this has failed in every single country that this line has been trotted out. So clearly, this is one of the major myths that we're all facing. And we're being misled about the fact that there's one diet that fits all. We're misled by the fact that it is simple calorie counting will work. We're misled the fact that exercise makes you lose weight. And endless other myths. So let's find out a bit more why this is wrong and what else might be behind it. We're told that these diets are crucial to us, and there's a new best-selling diet all the time, excluding something from our diet, and this is the secret. Just exclude that and everything else will go away. So let's take away grains. Gluten is deadly. Let's just take away fats, and let's take away carbohydrates. Let's take away anything that's unrefined starches. Let's take away dairy products, nasty dairy products that give you allergies and things we've never meant to have. Let's just take away, there's cheeses, let's take away meats, and all you're left with is maybe a glass of juice and lots of vitamin pills. Okay, perfect. You're bound to lose weight if you just have those. And this is the nub of the problem here. We're stuck with these extremely restrictive diets, and this is affecting us in ways we've never thought possible. Now, another big myth that actually started predominantly in this country and spread across the world like a disease was the fact that fat is bad for you. High-fat diets, bad for you. They give you heart disease. Fat equals fat. That was a very simple message. And everyone followed it. Every doctor followed it. They still do today. And this study came out 2 years ago, and it follows epidemiological evidence that the people that eat the most fat in Europe live the longest. These are the people in the southern Mediterranean. They eat 2 or 3 times as much fat as the average American. And they're the centenarians. They're the ones laughing in their retirement at age 100 in the sun. All that fat they're eating. And this study was the first randomized trial that lasted 5 years. It's been the best study ever done on nutrition. 7,500 Spanish people were randomized to 2 groups. And these were 60-year-old Spaniards who were a bit on the fat side, but no disease. So pretty average. And half of them were given the low-fat diet as prescribed by every American physician. And the other half were randomized to a high-fat Mediterranean diet, supplemented with extra olive oil in case they didn't have enough, and extra nuts in case they didn't have enough fat and calories. And what happened, the study was actually stopped early because an excess of deaths. And that happened to be in the low-fat group. So they had a third less heart disease, stroke, diabetes, and even a third less breast cancer. And this is the final nail in the coffin, I think, for the low-fat hypothesis. But this is one of these extra myths that we've been misled for so long, that what we're eating is not just a simple idea of fat, protein, carbohydrates. It's not this reductionist idea. There's much more to it. It's more complex. And there's an elephant in the room here, and there's also an elephant on the slide. And that is, we've been missing the most important organ in our bodies, that is our microbiome. All of us have 100,000 microbes in us that are 10 times more than we have cells in our body. So we are 90% microbe and only 10% human. And we're actually only about 1% genetically human because they actually have 100 times more genes, all of them pumping out chemicals and vitamins and hormones that we all need and keep us alive. And until recently, we thought they were just nasty things that we just had to get rid of with sprays and antibiotics. They were deadly because we could just culture a few of them. And it turns out that with genetic technology, we can now measure hundreds and thousands of the different species very precisely, and we know that 99% of them are good for us. They're our friends. And we've been trying to kill them for decades. And it also turns out, this new technology, that when you study people, and we've studied thousands of people now in the population, everybody is quite unique. We share only a small percentage of our microbes with the rest of the population, or even the person sitting next to you, even in our families. We all have a unique fingerprint that goes with us all our lives. And this is also explaining why perhaps one diet, one idea doesn't fit everybody, because we're much more different than we thought. Now, I've been studying twins, as you've heard, for over 21 years, and we have thousands of them, and we've started looking at their microbes as well. And as you know, when identical twins are born, they are genetic clones, exactly the same. But like any other babies when they're born predominantly sterile. No microbes at all, and at birth, those microbes get into the system and develop this community that lasts all our lives. And those first few days, months, are really crucial to forming these microbes that play such an important role in our lives. And it turns out that there are the major factors that alter this, that if natural birth, which by evolution for millions of years has meant that the baby's face goes through the birth canal and gets exposed to all the mucky bits that come out, is deliberate. And it all gets in through the mouth, and those microbes act as fertilizers for all the other things that happen and allow the baby to then digest breast milk, etc. And also build the immune system. Without it, babies would die very quickly and have no decent immune system or brain. Now, we now know that in the modern world, where everything is safety first, caesarean sections rule. And in the US, over a third of babies are now brought into life by caesarean section. And in places like New York and certain bits of Boston, it's over 65%. And this totally changes the microbes. And when we looked at twins, the fact that they can differ depends on who picked them up first in the delivery room. I was delivered by caesarean section, so I'm desperately trying to find out who that nurse was. I can blame her. So these things are very important. And twins generally have similar microbes as they grow up, as all other things, but then they can separate and be quite different. And so although we found, on average, there was a genetic component to having your microbes, there were individuals, twin pairs, were quite different, like this pair here, who were differed by about 30 pounds in weight and had very different microbial compositions. So it looks like the microbes inside them are playing a role in some of these metabolic differences. And we went on to look at over 1,000 of these, looking at the role between which microbes might be important in obesity. And we did this with our colleagues at Cornell University, who did all the genotyping and the hard work. And what it showed was, yes, there were certain microbes that if you had them, either made you more likely to get fat or less likely. And the one that the tabloid newspapers in the UK called the fat-busting microbe is this one, which most people can't spell, Kristensenella. I can't even say it. But it was a very unknown microbe that all of us have in tiny amounts, but a few people have a lot of it. And those people invariably were skinny. And it turns out that when you take the poo from somebody with this bug and you put it into mice who have made sterile, you will be able to keep that mouse skinny even if you feed it endless junk food. So people are now scouring the internet for this microbe to put on their breakfast cereal in the mornings. But some of you will have it and others won't. And this is just an example, the tip of the iceberg of what we can find by digging deeper into our own microbes and the whole world that's inside us. Now, what I've said for obesity needs to be thought of in a way of many other diseases as a garden. Basically, if you're healthy and skinny and you don't have any diseases, your microbes will be like a rich country garden. All our gardens in England are like this. Because it's always raining and it's always sunny. But if you have a disease, if you're obese, if you're diabetic, if you have any number of gut infections, colitis, autoimmune disease, allergies, irritable bowel syndrome, autism, you name it, you will have a disordered garden. You will have a barren garden that has less diversity of species, and in that barrenness, you get these nasty bugs taking over. And Eric is going to talk a bit more about some of those bugs taking over that whole system, which wouldn't be allowed to in your nice garden that's full of all these species which are protecting each other. Very important to bear this in mind. Now, cheese. I went on a 3-day intensive, unpasteurized French smelly cheese diet. I wouldn't advise it. Great for day one. Fantastic, because I was allowed 2 glasses of red wine with it. And I was enjoying my smelly French cheese a lot. But by day 3, really, I'd had enough. And I think my microbes as well also had enough. You can have too much of a good thing. But all doctors say us, don't have fatty cheeses. Very bad for you. You know, particularly those people with heart disease or on statins and things. It's a taboo area. And yet, people who eat regular cheese, all the epidemiology studies show they have less heart disease, less diabetes. And the French, our great rivals across the Channel, annoyingly live much longer than the Britons. And they eat 3 times as much cheese as we do. It's just not fair. So what are we doing about it? And what is it about cheese that's there? Well, if you take just a crumb of cheese, you're going to find billions of microbes in it. Billions. And these microbes, as you can't see, are mainly bacteria. But on top of that, there are also 5 times as many viruses. And they're fungi as well. If you take the Roquefort in those blue veins of the cheeses, they're all funguses that you're eating quite merrily. So we're eating this whole living organism when we eat cheese, and it's likely that these have effects directly or indirectly on our other microbes and keep us healthy in ways we don't yet understand. And for those who like to eat cheeses like this with crusts on it that are aged in the old traditional ways, you should know there are other little things on there called cheese mites, which burrow into the cheese and give it its flavor. And these cheese mites upset a few vegetarians for some reason because they get eaten every time you have your cheese. They're chubby little things, and they all taste just of cheese because that's all they're made of. But it's something to bear in mind when you do tuck in. But you may have to go out of your way to get it because the FDA, in its wisdom, has banned unpasteurized cheese. Now, after cheese, I wanted to then go and do a junk food diet, and decided I wanted to do a 10 days intensive McDonald's diet to really see if I could really improve my microbes through all that healthy eating. But after my cheese diet, I wasn't feeling quite up to it, and I thought I really need an expert to do this, someone who was really trained and dedicated, and he happened to also to be a student, which was good. He was hard up for money, and he also happened to be my son. So it all fitted. His friends were very jealous. I gave him the cash, sent him off to have all his meals at McDonald's for 10 days. He was delighted. But by day 3, the novelty was beginning to wear off, and some of that zest he had for life was going. And he said, "Dad, do I really have to carry on for another week?" I said, "I'm afraid you do." And by day 10, really, his opinion had changed, and he didn't feel so good at all. But what was really serious about this was that after day 4 or 5, we'd start to see a dramatic massacre of his microbes. He'd lost 40% of his diversity in his species. And this is getting rid of over 1,200 of these species types that we'd seen. And the conventional idea was, well, that's just high fat and high sugar, bit of salt. These things must be upsetting his metabolism. But actually, that probably isn't the case because that doesn't really affect the microbes. The explanations are that he was getting very little fiber for the microbes to feed off, except that little bit of gherkin you get, that little tiny sliver, which might be the only bit of fiber you get in a burger. But it could also be the other products you get in processed foods, the emulsifiers, which we now know affect our microbes and can distort them. All these things in the mayonnaises and the tomato ketchups, plus also preservatives and sweeteners. All of these things are bad and affecting him. Now, what else can you do to help your own guts? Well, obviously, eat more greens. That's the standard treatment. But knowing why is important. And we now know that there are certain greens that are better than others. Lettuce doesn't really help you very much. But if you take these root vegetables, things like chicory roots or artichokes or leeks, onions, garlic, those are the basis of the Mediterranean diet. They are packed with things called inulins, which microbes absolutely love. And they produce lots of helpful chemicals that help our immune system based on them. Then you've got other things that also are rather strange. We've been told are bad for us, like peanuts and assorted nuts and seeds, and even coffee and dark chocolate, full of sugars and things, red wine and olive oil. What unites them and why are they good for our microbes? They contain polyphenols in huge amounts. They are superbly rich in these polyphenols, and again, this is food for the microbes. The microbes feed off all the different types, and just in red wine alone, there are nearly 100 different polyphenols, and about 60 in extra virgin olive oil. Not the cheap stuff. You've got to get the expensive, good stuff. Cold extracted, extra virgin olive oil, you get the maximum polyphenols. And if you do that, you're giving them a huge treat. Then we've got yogurts and fermented products. Obviously, they're good because they contain microbes. But are probiotics good for everybody? Well, they are if you're a rat. All the mouse studies, all the rodent studies show absolutely that virtually every single test, they're fantastic. They can prevent anything. The data in humans, unfortunately, isn't quite as good. You've got to be very sick or very old or very young to really benefit, or be on nasty antibiotics. And Eric's going to talk a bit more about those. So yes, it works, but if you're really healthy, they don't seem to have a great effect at the moment. Antibiotics, we've got to talk about them, and there's a great book on this subject by Marty Blaser. Some people believe that the epidemic of obesity in this country is due to overuse of antibiotics. And in the US, they're overused more than pretty much any country in the world. Average 18-year-old has about 18 courses of antibiotics. Average 3-year-old has had 3. 3/4 of the time, totally unnecessarily. And each time, it's like a nuclear bomb going off in your gut. And you have enough of them, you wipe out all the good guys. And this is where you get problems. So avoiding those is really important for our future health. But also in the food we eat, because much of the food we have is contaminated with antibiotics anyway, and as is our water supply. What do you do if all this fails? You've tried everything, and you've still got rotten microbes you can't get rid of. Well, the microbial makeover is the ultimate solution. And these could be the tablets that help you. We call these technically poo capsules. And they are frozen poo. And basically take 10 of these or 30 of these, you will be cured of serious infections. And people are now looking at this much more seriously, and there's now 500 centers in the US offering these transplants for serious diseases, of which 14,000 people a year die in the US. So this is now a mainstream treatment. And Eric is at the forefront of helping this system get around the world. And as part of this new interest in the microbes, people are testing their own microbes. And the British Gut Project is an offshoot of the American Gut Project, which my colleague Rob Knight started a couple of years ago. And it's a citizen science project where anybody can go online, look up American Gut, make a small donation in both senses of the word, and get their results back, and also contribute to a huge database telling about not only the American gut, but all the British gut, and hopefully the global gut, about what's normal and not normal. And we see this amazing variety. So do go there, and you can get your own, you can compare yourself with other people, which is what people want to do. So to summarize, we should all treat your own bodies like you would this precious garden. And that means looking after it carefully, understanding that everybody's soil is different in all your gardens. And treat it with good fertilizers. You sprinkle on nice products every now and again, add new seeds to it. You change the soil, you add variety, you eat seasonally. You give it, above all, care so that you avoid toxins, those nasty antibiotics and other things that we've been, processed foods. And above all, the key is diversity. Eat as much diverse foods as you can. You will have a diverse garden and you'll stay healthy. And the other thing to remember is that if you do that, you've got to realize that with diversity, never again will you eat alone because you have 100 trillion friends in there as with you. Thank you very much. So I'm going to hand over now to my colleague, Eric Alm from MIT, who's going to take the baton on and give us into the next level. Thank you, Eric.
Well, thanks, Dr. Albright, and thanks to the Museum of Science for inviting me back. It's really honored and really humbled to kind of share this stage with Tim. I want to talk to you about basically taking the microbiome and thinking about, because I'm at MIT, we're engineers. We always think about, here's some new science. How do we engineer it to improve the human condition? And so one of the things that I always ask people when I give this talk is, why are we talking about the human microbiome in 2015? Was there some new technology that was invented on the microbiology side of things? Did we learn how to culture more of these organisms? Was there a new microscope that was invented that allowed us to recognize the importance of the microbiome? Does anybody have a guess? So yeah, so Clostridium difficile, so there's some clinical information that says it's important, but I would argue that the field of microbiology started with infectious disease roots. And so we've had that on the forefront of our knowledge for over 100 years. So what is it about 2015 and really maybe the last 5 years that's sparked this interest in the microbiome, and most of the microbiome being these healthy bacteria, the ones that aren't really germs? DNA sequencing. So a few years ago, we decided that if we could only sequence the human genome, we would reveal all of the drug targets. At that time, we didn't know if there were 5,000 or 100,000 genes in the human genome. But once we had those precious drug targets, they would each be the target of a new drug that would impact human health. And so as soon as we figured out how to sequence the human genome, it was a $3 billion project. We got the first human genome. We waited for those drugs. And we put a lot of work into developing the technology and making it very high throughput. And during the process, we got really, really, really good at sequencing DNA. Okay, so it's hard to see here, but this is a logarithmic graph. Okay, so if I follow this line, that's going to be similar to Moore's law for computing, and it says that every I think a couple of years the computing power doubles for the same price and the same amount of electricity and things like that. This is a logarithmic graph, so you can see that this is growing faster than exponential. Like very few processes obey laws like this. Very few things grow faster than exponential, and I don't think it's slowing down, which is amazing. So not only have we been able to exponentially increase our sequencing power, we're doing it faster than exponential. We are so good at sequencing, we should have all the drugs we will ever need within the next few years, right? This is the number of $1 billion of research required to produce one new FDA-approved drug. And the time axis here is the same, and this goes up to 2010. I'm sorry, the inverse of that. So this is the number of new drugs that we're getting for each $1 billion of research that we put in. The graph is going in the wrong way. We got really good at sequencing that DNA, and the drugs haven't come. To some extent, maybe we were over optimistic about our ability to take that complex genetic knowledge and turn it into new drugs. I, for one, am very confident that soon, as we start to sequence more humans and figure out what the action of each of those drug targets are, that we are going to be able to see that explosion in new drugs. But it's going to be a few years out. And what I'm going to argue to you today is that the microbiome offers a really unique opportunity. And that opportunity is that we can leverage the fact that we're really, really good at DNA sequencing and use that expertise to develop new drugs that are based on microbial microbial species that are in our guts. So it's the idea of bugs to drugs, okay? And I'm going to explain essentially how I think this is going to work. This is an oversimplification, okay? But essentially, we're looking at healthy humans, and we want to culture all of the bugs that live in and on those human beings. This is a culture plate. Each one of those little dots is one bacterial isolate. We want to take those isolates, put them in the freezer, make master stocks so we can always go back and we can find that organism, we can grow it up. We can study it in the lab, or maybe we can put it in a pill and feed it to a patient. At the same time, we're taking that same material and we're getting the DNA sequence. We're using that to identify the bacteria, and then we compare the microbiome, the bacterial content of healthy groups of people and sick groups of people. And if we find that the sick people have a very similar microbiome to the healthy people, except they're missing one particular drug, now we have a hypothesis that we can test in the clinic. We can take that species that they're missing, we can take it out of our freezer, we can put it in a pill, and we can feed it to those sick people, and we can see if they get better, right? And so this is a very short circuit from DNA sequencing to a clinical trial, okay? That's very naive. We probably wouldn't do it just based on that. We'd do a little bit of background work before we entered into a human clinical trial. But that's the basic idea. So looks like the mic is out, but I'll try to speak a little louder so you guys can hear. So this is the idea. This is the sort of new paradigm that we imagine in drug development that could result from the emergent science of the microbiome. So what are the diseases that microbiome is associated with? This is a very, very non-comprehensive list of a number of different diseases that have been associated with the microbiome. Oh, do you want to change the battery? All right. Yeah, you can take that. So this is a very short list, and one in particular I'll talk a lot about today, and that's Clostridium difficile infection, but I'll get to that in a moment. But just fair to say that there's a whole host of diseases that we know are associated with the microbiome, but sometimes an association doesn't mean causation, right? Two variables can be very correlated with each other. It doesn't necessarily mean one is causing the other. Okay, so we'll try the speaker. So if it turns out that most of these associations are just correlations and not causation, that's fine, especially if a few are, then maybe we'll get drugs for a few of these diseases, which would be fantastic. But when we can't establish causation, in many cases, all is not lost because what we have is a diagnostic. We have a molecular diagnostic, and a lot of these diseases that are associated with microbiome don't currently have a good molecular diagnostic that we can use in the clinic. Oh, perfect. All right, that's great because Tim told us we should get exercise, so I don't want to kind of stand behind the podium all night here. So how would this work? Well, here's one case that's really close to kind of the research that we're doing. So this is a fictitious student. She's a 20-year-old student at MIT. Comes into my office, says, Eric, my grades are suffering. I tell her to go see the doctor because she's having nausea, weight loss, depression, a slip in grades. So she goes to the clinic. Tells them about her symptoms, and the doctor is now thinking IBS. So IBS and IBD are very different diseases with very similar symptoms, okay? But very different treatment regimens. So the doctor's thinking IBS because she doesn't have any of the external, extra-intestinal manifestations that we usually expect for IBD. Then finds out that her father had Crohn's disease. Crohn's disease is one of the different variants of IBD. So now the clinician is thinking maybe it's Crohn's, looks at all the information and really doesn't know one way or the other, so she needs a colonoscopy. Well, turns out that her dad was one of the rare cases of folks who come into the clinic, get a colonoscopy, end up with a punctured colon, and so that was a huge ordeal. She absolutely doesn't want that, okay? So she refuses to get the colonoscopy. but her grades are suffering, and our MIT students really care a lot about their grades. Okay, this is a big deal, maybe bigger than the other symptoms. And she wants a diagnosis, and she wants a treatment plan. And this is not like a completely hypothetical case. This is something that could be fairly typical that we might see in the clinic. And currently, we don't have a lot of really great non-invasive diagnostics that can really compete with colonoscopy and get Ashley sort of the diagnosis and the treatment plan she needs. What do we imagine in the future? Well, we imagine sequencing her gut microbiome and trying to figure out, is this a microbiome that's telling us, maybe Ashley has IBS, or does it look like the microbiome of someone with IBD, if this is a microbially associated disease? And so we imagine like bringing out these diagnostic tests and then taking that very big data, right, that a clinician's not going to sit there and plot and analyze and putting it into an electronic medical record system that doesn't just track all of the symptoms and what the result of the lab test is, but a medical record that actually makes predictions. It makes predictions, makes a diagnosis, and then gives a probability of remission if we treat with this drug or that drug, and makes recommendations based on all the genomic data that we're putting in. So that's kind of the future that we envision microbiome really contributing toward. How close is this future? I would argue it's not very far off. So I'll tell you a little story about a project that we started, and this is a project that really drew me into the field of microbiome. So Athos Bousvaros over at Children's Hospital came to me and said, Eric, we've got a problem. At this time, Athos ran the pediatric inflammatory bowel disease clinic over at Children's Hospital, and he said, we've got a problem because on average, there's a 3-year lag time before the onset of disease, onset of IBD in children, and accurate diagnosis. And what that means is that there's 3 years go by before symptoms show up and patients get the treatment they need. And oftentimes that can lead to poor outcomes. So oftentimes the first intervention has to be surgical, okay? And these kids have to have part of their colon removed. Whereas if we'd gotten them earlier and managed disease, maybe we could establish a better outcome. So he said, maybe there's some way that we could get a non-invasive stool sample, sequence it, and determine if the patient had inflammatory bowel disease or maybe just IBS, something like that. So the other option, of course, the gold standard is a colonoscopy. But the downside here is every 8-year-old child that comes into the clinic, you don't want to send them out for colonoscopy, right? Colonoscopy is not very pleasant. And so there's this fundamental trade-off, and Athos said, maybe we can do better with microbiome science. So what Athos did is he went out and he took a good sample of folks that came to his clinic that he later diagnosed as not having inflammatory bowel disease. So he did a colonoscopy, ruled IBD out. And then he recruited some patients that had inflammatory disease, okay, in 2 different versions, Crohn's disease and ulcerative colitis, the 2 main forms of IBD. And that's what you're seeing here. So there's a lot of data, but I'll kind of walk you through it. So every column in this graph is a patient. It's a sick kid, and these are the control kids. They're still sick. They're showing up like Ashley did, right? They maybe have weight loss, depression, grades are slipping. And they're showing up to the clinic, but Athos later determined that they did not have IBD. These are the children that had Crohn's disease, and these are the children that had ulcerative colitis. And on the rows here, what you see are all the different bacteria that we sequenced in their guts. And so I want you to look at this and tell me if you see any bacterium that is really indicative of disease. Like if they have this bacterium, they're sick. That's the germ. That's the one we need to get rid of. The Clostridia, I actually see more in the controls than I do in the sick kids. So I think these kids want Clostridia, but I still see it in the sick kids. So the only bug that's more abundant in the sick kids than the ones without IBD is E. coli. Okay, so all of the kids that had a lot of E. coli had inflammatory disease. But if you're to use lots of E. coli as your biomarker for disease, you'd only detect disease in about 10 or 15% of the kids. So on its own, it's not good enough. So there's not a key hallmark change in microbiome that tells you whether a child has inflammatory disease or not. But what we were able to do is we were able to take all of these weak signals, all of these things that look like, oh, you know, maybe if they have a little bit less Clostridia than you would imagine, maybe they have disease, and take all of those weak signals, combine them together with machine learning, and we actually built a very good predictor that we later found was better than the current gold standard in non-invasive tests, which is fecal calprotectin. Okay? So I would argue that this future is not very far off, and one of the things we found in this study is that when we reanalyze the data, and as we put more and more patients into the model, the predictive ability of the model, the accuracy of the model got better. So it's a diagnostic that the more times you use it, the more accurate it gets. And that's one of the really exciting things about microbiome-based diagnostics. So we don't just want doctors to have access to this data. We want to get this data, this non-invasive data that we might be eventually collecting daily with smart toilets, or we're thinking about systems with smart wipes that patients could use that will give the information back to clinicians. We want to get it back to patients. And so we want, if we diagnose Ashley with inflammatory bowel disease, we want Ashley's cell phone to buzz 2 weeks before she has a flare-up and say, Ashley, there's something going on. You need to maybe see your doctor now because you're not sick yet, but based on your microbiome, we think you might be getting sick. And that's sort of the direction that we're heading. I would argue that this future is not very far off, so I'll tell you a little story about a self-experimentation study that I think Tim alluded to. So my student Lawrence David and I, in 2009, decided that we'd track our microbiome, and we would also track 300 different parameters that corresponded to our daily life and behavior. So we wrote down every morsel of food we ate, we wrote down how much sleep we got, you know, our sickness and any medicine we took, all of our travel, basically everything we could think of. We recorded it for about a full year. Lawrence said, if I write an iPhone app to track that information, can I charge an iPhone to the grant? I said, sure. So now there's an app for that for anyone who's interested. And so this is what we found. So in 2009, this is my microbiome in 2009. This is Lawrence's microbiome in 2009. And the thing I want you guys to notice is they're very distinct. Okay, so there's a lot of variation day to day, but I could pick any day from my time series and show it to you, and you wouldn't need any fancy computer algorithms to figure out if it was my sample or Lawrence's sample. So our microbiomes are very, very distinct. They're very distinct ecosystems, and they change day to day, but they maintain their sort of unique quality that's different from each other. Okay, so what are the things that affected it? Certainly diet played a big role. But one particular life event really affected mine. So about 3/4 of the way into the study, actually, I would say almost 6 years ago to the day, I took a colleague up on an offer to go out to brunch. I made the mistake of ordering the chef's special French toast, and 48 hours later, I was deep in the throes of a horrible salmonella infection. And you can see the salmonella down here. So this is my microbiome again. And here I've blown it up. So every single row is a different bacterium. And the red colors mean there's more of that bacteria on that day than other days. And the blue colors mean there's less than on other days. And you can see as soon as the salmonella appeared, all of these bacteria basically disappeared. And about a week after infection, some of those bacteria came back, but the majority went permanently extinct and were sort of extinguished from my microbiome, but they were replaced with all of these bacteria in here. And so we saw complete restructuring of the microbiome as a result of this life event. And what I think is fascinating is even after that total restructuring, you can still tell it looks like me if we go back to the other picture, right? So here's the event. It still looks like me. It doesn't look like Lawrence. So my microbiome was able to completely reboot itself, but reboot sort of a different operating system than Lawrence had. And that's still, I think, one of the mysteries from that study that we still don't fully understand. So we know we can start to monitor these things. The technology is getting cheap enough that we can really roll it out, and we're thinking of doing that next year for inflammatory bowel disease patients. Microbiome is only the beginning of the things we want to measure, so we want to use wearable devices and a lot of technologies that are making this whole process quite a bit easier. We want to look at the immune system and see how the body is responding to those microbes, not just look at the microbes. And so that's what we see as kind of the future. I want to talk about one more thing. So we talked about diagnosis, but I didn't tell you, I said I want to make drugs from bugs. How do we do that? And the best model for doing that we have right now is looking at Clostridium difficile infection. And so we want to move from diagnosing to engineering the microbiome, but let me tell you a little bit about the course of disease first. So what we have, we generally have a person with a healthy gut, so all these germs and stuff, that's good, okay? You guys should know that by this point in the presentation. So this is a healthy microbiome. We hit it with antibiotics, and we sort of clear-cut that beautiful forest or that beautiful English garden. And so now we have all of this empty niche space that invasive species can come in and get a foothold in. Okay, so if we deliver antibiotics and the patient is exposed to C. difficile, that C. diff can basically take over. And once C. diff takes over, what do? We give the patient antibiotics again, of course, because now they have a terrible infection. In many cases, the antibiotics will return us to this state, in which case the patient will become reinfected with C. difficile. Then it becomes even more likely that the next time we treat them, and what do? Of course, we give them antibiotics again. It becomes more likely that they're going to recur with C. diff. And after 3 or 4 or 5 cycles of antibiotics, it starts to become very unlikely that the patient is ever going to respond to another course of antibiotics. Okay? So they're sort of locked, sort of alternating between living on vancomycin, which is not fun, and having C. diff, which is less fun. Okay? And so 2 very terrible choices. But it turns out that there's a cure. And the cure is to treat the patient with antibiotics, and then instead of waiting for the patient to be exposed to C. diff and recur, we give them a fecal transplant. So we take bacteria from a healthy donor, and we infuse that into their gut ecosystem, let those healthy bugs take over, and they sort of crowd out we replace the clear-cut forest with the beautiful, lush English garden, and then C. diff can't get a foothold and is effective in 85 to 90% of the cases. So we got involved in a study at Mass General. This was a study where there were 4 healthy donors that treated about 20 patients, and about 18 of those patients recovered, okay? And especially one of those donors treated 12 patients, okay? And this was a study that was carried out by Libby Homan, Ilan Youngster, Jenny Sauk over at Mass General, and Dirk Jeevers and Bruce Byrne at the Broad Institute. And we used this. So we went in and we looked at the clinical study, and we say, we want to get some genomics. We want to see what's really going on with those microbes. And so we did that. And so a really great graduate student in my lab, Chris Smiley, who's a computer scientist, he made this graphic of what a patient looks like, what their microbes look like, just coming off of vancomycin. So this is a patient with Clostridium difficile, okay? And so each one of these blue bands is a different bacteria, okay, in our sick patient. Down here, you see, these are all different bacteria that were in the healthy person that gave bacteria to the patient. And you can see they're very different. And now I want you to try to squint and see if I add these bacteria to this patient, what's going to happen? And this is what happens. So this is our patient after the patient gets a fecal transplant from this donor. And we squinted at that for a long time. It doesn't look like just adding the 2 profiles together. It doesn't look like an AND gate or an OR gate or any of the different things that we could think of. The human eye is pretty good at pattern recognition, but we didn't see any patterns here. But Chris went back. He built a machine learning algorithm to try to solve the problem of, given the patient and the donor, what are they going to look like post-fecal transplant? Whoops. And this is what he found. So he found his algorithm was about 86% accurate in actually predicting what the patient would look like after the fecal transplant. And the thing that's very exciting to us is that now it says, if we know what the patient is supposed to look like, and we know what they look like now, and we have a lot of different possible donors to combine them with, then we can sequence our patient, we can sequence all the different donors, and we can ask our computer algorithm which of the possible donors that we could use to give a fecal transplant to this patient will transform this patient into what we think the ideal healthy gut would look like. So the only other step that we need to do that is we need a bank of hundreds of different healthy donors that we could just call up and say, yeah, give us donor number 51. So it turns out a few years ago, a student in my lab had a family friend, and this was a young guy. Most of our C. diff patients they're geriatric patients, and so they're a little bit older. This was a young guy who was a Princeton grad, had his whole life ahead of him, got a terrible round of C. difficile infection, got multiple recurrences, couldn't hold down a job, was really ruining his life. He was an insider in the healthcare industry, so he knew about fecal transplants, and he knew if he could just get a fecal transplant, maybe he would get that 90% likelihood to be cured. He called around. He heard that some people were doing this as part of research study. Nobody really was doing it. Nobody had availability. Eventually resorted to giving himself a fecal transplant, and eventually was cured. But the whole ordeal took much longer than it should have, and he had to practice a potentially very dangerous medical technique, not under medical supervision. So a really fantastic graduate student in my lab, Mark Smith, said, this is unacceptable. We know how to make this stuff in the lab. And so for about a year, we shut down all other activity in the lab so we could manufacture pharmaceuticals in the corner of the lab. We didn't tell MIT. They would have shut us down. But now they're happy because the result was really amazing due to all their hard work. And so eventually spun off into a nonprofit organization that today has about 36 full-time employees that ship fecal transplants around the country. So Tim mentioned there's about 500 facilities treating folks. 430 of them are within our network and receiving product from OpenBiome. And that means that about 94% of the U.S. population is within about a 3-hour drive of one of our clinical partners. And so we're really trying to provide safe access to this product, serving about 49 different states. About 7,000 patients have been cured so far. And one statistic that's not on here that I think is really phenomenal is, based on our internal estimates, we've saved the healthcare industry about $100 million in the first year or two of operation. And so I think I'll sort of end there, and I want to note that there's a couple folks from OpenBiome in the audience. I'm sure they might come up afterwards if people have questions and tell you more about their nonprofit operation. So I want to thank some of the folks that contributed, and this would be a good time. I have a few questions for Tim, so maybe if you come back up here, we could start the conversation part of the evening. So Tim, I was really pleased to see the self-experimentation, something that I'm also a big fan of, and so I thought we could start out with sort of a fun question.
No one should share a refrigerator with us guys. That's it.
Absolutely not. What's the most exciting experiment that you've personally participated in related to either diet or microbiome or anything really?
Very exciting. I had to abandon one, which was I wanted to really do the intensive olive oil diet. I'd heard that fishermen in Crete used to drink a glass of olive oil for breakfast, and they lived to be 100, and it was all fantastic. So I said, well, I'd never actually met anyone who'd done this or actually seen it. So I said, well, is it possible? So I got the best grade Spanish extra virgin olive oil, and I sort of built myself, and I said, well, in a week I've got to have half a liter of olive oil, and I just wasn't getting through it with the salads. So I said, I've got to start drinking it in a glass. So I made the mistake of having a glass on an empty stomach, and then I went off to get my hair cut and then fainted. So I decided that some things were just a bit too dodgy for me to do. So, but yeah, and I had a colonoscopy and saw what happened to my microbes before and after. and not much happened, actually. I was very disappointed, but it was an experience, you know. And then the cheese you know about, and other ones. But yeah, it was fun. But you travelled around. You went around the world with your one, didn't you?
So Lawrence went to Bangkok. He went to Bangkok, and his experience was a lot different. So his microbes changed almost as soon as he got off the plane. He picked up a cocktail of about 3 different enteric pathogens. I had to Wikipedia one of them. I had never even heard of it before. But his microbiome completely shifted. But what was really interesting is as soon as he got back to the US, it came right back. And so that was really different than what happened to me with Salmonella, where it was more of a permanent change.
But I've heard it said that it's a cheaper way of getting a fecal transplant is to just go to India for a couple of weeks.
Well, the thing
and get
about
a reboot. So,
the thing
would you
about
recommend
fecal
that?
transplants is you probably don't want the cheapest one.
No. I guess so. But if you went 20 times, eventually you might strike lucky.
That's right.
So where do you see fecal transplants going? I mean, if we were talking 5 years from now, what do you envisage? Because at the moment, Clostridium difficile, it's accepted because it's killing people and it's a life-saving procedure. And yet many people on the internet are discussing doing it themselves for more minor conditions. And yet the data isn't that great at the moment. But where do you think what will we be treating it for, do you think, in 5 years? What would you guess?
Yeah, so I think 5 years is such a crucial time. So it's like right now we've got the technology to look at microbiome. We also now have the infrastructure to do fecal transplants, and for example, OpenBiome is releasing pills soon. So it's becoming very easy to conduct these trials. And I think in the next 5 years, what we're going to do is we're going to look at all of the different diseases that have an association with the microbiome, and we're going to find of those, dozens, maybe even hundreds of diseases, which are the 5 or 6 where microbiome is actually going to cure things, where it's going to work like a charm, just like C. diff. And so I think in the next 5 years, we're going to try fecal transplants and try these very crude interventions for lots of diseases, and we're gonna narrow down on what are the diseases where it has a chance. And then I think the 5 years after that are going to be spent saying, we know fecal transplants kind of work. They probably won't be as effective as they are for Clostridium difficile. I think that's going to ultimately prove to be an outlier. But when they work a little bit, then we're going to think about, okay, can we make a synthetic cocktail of drugs? And some of those organisms in those drugs might be engineered to have even stronger phenotypes and treat disease even better. And so after 5 years, we'll maybe narrow the list of diseases we care about. But right now is really exciting because we don't know what disease is going to respond. And so at least at our microbiome center, we're trying to have a really diverse portfolio. We have a focus in IBD, but we want to have our hands involved in many different diseases.
And when you have a bone marrow transplant, generally you get genotyped and you matched for your HLA system with a donor. And until recently, we thought that wasn't important in the microbiome, but twin studies that we did with Ruth Lane Cornell have shown quite conclusively that the genes do play a part, and our new data is suggesting it's even stronger than with the 16S data. So in a way, your best donor is going to be yourself because you have the same genetic background. So do you think we should be setting aside our poop when we're nice and healthy and young, stick it in the freezer, and then get it back again later in life, maybe to keep us forever young or healthy? What about that as a future? Do you see that as a...
So, yeah, self-stool banking.
Well, people are doing it for stem cells in their babies. They're paying their money at birth.
That's right.
So maybe, when we think we're at our healthiest, wherever that is, that's when we should be doing.
One of the things, and it brings up an interesting question because there's a very confusing set of regulations around fecal transplants right now, and basically any sort of microbial-based therapeutic right now. By those regulations, you can bank your stool today, but you can't get it out of the bank. You can't get it out of the bank unless you have recurrent Clostridium difficile. And so it might be a good idea to bank your stool before you take a round of antibiotics. And then if you get an infection, you'll get the antibiotics to get rid of the germ, and then you go back to the freezers and repopulate with your own bacteria. Or maybe for me, it would be after Salmonella, I go back and repopulate.
What you're saying is you currently wouldn't be allowed to.
I can put it in a bank.
You can't put your own stuff back.
And the FDA won't allow the bank to give me my own poop back. Unless I get a recurrent C. diff infection, but that's not worth it.
So, and the analogy would be if you spit into a tube, freeze it, the FDA wouldn't allow you to have your saliva back in your body. It would be treated as a drug. Is that right?
You'd have to ask the FDA that. I don't put words in the FDA's mouth, but
okay, it sounds kind
I
of
can
crazy.
definitely tell you that at a stool bank you wouldn't be allowed to give that material back.
So the whole thing has thrown up this whole problem about, what is our microbe? Is it ours? We say we're only 10% human. We carry it around with us. If it is ours, does it not belong to us?
Right.
It belongs to the institution that decides whether we can have it back again. So is it a device? Is it a tissue? Is it a drug?
Right. And so I've argued that it's a tissue product. It should be treated as a tissue product, and if you run a stool bank, you should follow regulations that are similar to running a blood bank. But the FDA has said very clearly that it's a drug. Poop is a drug. So it's a very weird stance to take, and it's leading to these sort of very weird situations where, yes, if you give me your poop and I put it in my freezer, I can't give it back to you until you get infected with C. diff, right? And so I think we definitely need to rethink the regulations around stool if we're going to use it as a medical product.
But hopefully in 5 years things will have moved on and people will have come to grips with this and treat it properly.
Right.
And you think in 5 years we'll be having a much more personalized service, tailor-made, working out what the exact bugs are, going to these databases, pulling off the one that fits both the disease and the person, and have a much more rational approach to this. That's your hope.
That's the dream. Yeah. Yeah, exactly.
And we'll have special yogurts that are also made perfectly for us to live forever.
Definitely a big fan of yogurt.
Big fan of yogurt. Okay. Well, on an optimistic note, I think perhaps we could open up for some questions
I think so.
From the audience. I'm sure we've whetted someone's appetite.
Here's a question right here.
Thank you very much. Very enlightening. My question is about the use of the bacteria for cancer treatments, such as the BCG.
So I think, what are the use of microbes for cancer treatments?
Yeah, I'm just looking into the future.
Okay. Well, there's a paper coming out soon that I've been told about that does show that chemotherapy can be altered by your gut microbes, so that it's likely that some of these transplants or special antibiotics will be used in conjunction with chemotherapy to improve the response to the drugs. So this is very hot, exciting news. So it looks like, again, for another life-threatening illness, by manipulating our gut microbes, which produce these vitamins, these hormones, these immune suppressant chemicals, which are really crucial in fighting cancer, that these could be another use and potentially another use of these fecal transplants. And why perhaps banking your own healthy stool for when you might get cancer or you might get infections could be the big thing of the future. Do you?
Yeah, I think we certainly know that the immune system plays a very big role in cancer, and we're seeing therapies that are based on modulating the immune system come out. So the fact that the immune system really is primed and programmed by the microbes lends a lot of plausibility, I think, to the idea that you could modulate things like chemotherapy and cancer therapy with by changing the microbes. But I don't know about the study that you mentioned, so I don't know of any work yet on that topic, but it certainly seems very plausible.
Yeah. Okay. Who's got the microphone? Okay.
Hi, thanks very much for the talk. Very interesting. So had a question regarding Crohn's disease in pediatric populations. I understand that there's been a lot of work with taking a look at their microbia and so forth. And one of the things you mentioned is about altering your diet and how that changes the microbial environment. Has there been any correlation with changing diet and success with treating Crohn's disease?
Yeah, so diet can be very, very effective. So there's enteric feeding. There's recently a trial that showed that it's basically as effective as some of the strongest.
This is feeding with
molecules.
this is feeding with a tube where you liquidize everything, and so you know exactly what's going in someone who's rather sick and can't eat normally.
Right.
So that you can get extra fiber and extra fertilizers for the microbes in this way. And
it's very, very efficacious. So we know diet can have an effect, but enteric feeding is it's pretty extreme. So you couldn't do it every day, right? And so I don't know that there's a lot that's been shown in terms of diets that you could stick to, like Mediterranean diet and things like that. In their role in suppressing IBD, but then again, I think there's less work on that topic than there are on more sort of molecular approaches. So it's also very, very, very difficult to track diet. So you can do a diet study where you do an intervention, but by logistical constraints, that's going to be a relatively short-term intervention in most cases. And then if you try to track diet via questionnaires and things like that, they're notoriously inaccurate. So you need to have extremely large cohorts to kind of get the signal out. So it's difficult to study, but I suspect diet is pretty important.
Okay, we have our next question over here.
Emmanuel, I understand that when you're sick, you have very little choice on what you take. Have you done any study in anal administration of the transplant?
So giving the transplant through the rectum? Yes. So it's either given through a nasal tube down through the stomach, or it's given through an enema or through a colonoscopy. So it can go either way. So increasingly, it's being used through the bottom, and that seems to be the route that's gaining more approval. So you're right. When people are very ill, that seems to be a route. But either seem to work, as well as these capsules.
Next question right here.
I was just wondering whether, in consideration of the significant rise of food allergies that we see now in kids that we didn't see in the population years ago, whether you've seen any connection between this and changes in the gut microbiomes, and specifically even within the same families through which birth canal several children have been born. Have you looked at that at all?
So I haven't looked at that personally, but studies are starting to come out, especially in mice, showing that you can transfer some of these food allergy phenotypes through microbiome. So those are pretty early studies, and we need to see them confirmed in humans and repeated and things like that. But there's certainly some promising leads on that front.
Epidemiologically, lack of diversity in the diet has tracked the increase in food allergies. And also the lack of reducing fiber in the diet has tracked food allergies. So the more people are worried about food allergies, the more restrictive their diets are. Like saying, there's peanut. I've heard of someone with peanut allergies. I'm going to avoid peanuts, particularly in pregnancy. That actually increases the risk of the kids getting peanut allergy and other things. So it's the more restrictive people are going, listening to all these wacky diets, cutting out grains, worrying about gluten, all this, they are causing problems. And these animal studies are confirming that, because you can reverse allergy in the kids. They've done it more with asthma than with food allergy. It's very hard to do food allergy properly in animals. But by giving extra fiber to the pregnant mums, they can actually eliminate the allergies in the kids. So I think we need to be realizing that it is a general trend. Nothing else really explains this massive increase in food allergies in the last 20 years. Remember, the first case report ever was in the 1970s. And you could tour the world on that one case report as a celebrity if you had a food allergy. We forget quite how common it's now been. And I think we have to redress and realize that the more microbial exposure we have to our kids, antibiotics, having pets, dirt everywhere, these are good things, as well as huge diversity of high-fiber foods, then we can start to reverse this trend. But,
Next question over here.
Hi, that was actually a good segue because I was going to ask, to what degree do you think your microbiome is affected by external things versus things that you actually ingest?
One for you, Eric.
So let's see. So you mean how has it changed, or how is it like inoculated from the?
Well, so how is it determined? So, are you just as likely to have your microbiome be changed because you touched something versus because you got food poisoning at a restaurant one day?
Right. So the kind of changes that we see. So in the data that I showed you on myself. What you saw is that I got food poisoning, and then everything radically changed. And there were totally new species in there, right? The same types of species, but totally new ones that I had never seen before. That's actually quite rare. So the normal day-to-day variation is generally a change in the abundance of the species that you have. And there's studies showing that, apart from these events like food poisoning that actually change what species those are, the species that you have can stably colonize for decades or, potentially even your entire adult life, right? So what we see is the external events, the behavior and the diet are really changing the abundances of the microbes that you have. And and it's these more rare events where something actually is ingested and kind of blooms within the gut. So I would say it's sort of can happen either way, and it normally just affects how much of each bug there is. And the biggest factor is diet, and within diet, the biggest factor that we were able to track was fiber.
We have another question over here.
You said that you ruined your son's microbiome. How did you get it back?
I often get asked this. He hasn't been keen to be in more of my experiments since then. So, well, I did offer him some salads and things afterwards. But seriously, 3 weeks later, we did test him, and he was still way below his initial starting point. So they hadn't recovered 3 weeks after. We've sent off some other samples, which would be a year after, to see if he's recovered, but haven't got those back yet. But I suspect that, if the insult is enough, so 10 days of zero fiber and all these preservatives, emulsifiers, you will lose some species permanently. And I think that's sort of a test, or they're below that level of detection, and you can't be encouraged to come out of the cracks again. So it is a worry that, it sounds like you can have the odd burger every now and again. That probably won't affect you too much. But if you really have nothing else along it as well as it, then you're in trouble. And there's this idea that, you can have some bad things to eat as long as, and there are some experiments in animals now showing that if you have some very high-fat junk food diets, but at the same time you have lots of fiber with them, you're much better off. So I think we can, in a way, safeguard ourselves against some of these problems by increasing a variety of fiber, not just one type of fiber, but a whole range of normal natural fibers.
We have another question over here.
This is probably way too complicated to answer tonight, but my daughter has an autoimmune disease. It shows in alopecia areata, and for a while she saw a huge effect of changing her diet, but then it kind of just stopped being effective, the change in diet, even though she continues it. What is the correlation between the microbiome, is that how you say it, and autoimmune diseases?
Yeah, so there's a very deep association between many autoimmune diseases and microbiome. I've even heard case reports connecting microbiome to alopecia. So it's certainly in the sort of sphere of things that can be influenced by the microbiome. And we think really the connection is that the microbiome is in the gut is sort of intimately interacting with the immune system, and then that immune system has effects that are more systemic. So there's interactions that are happening in the gut that aren't necessarily related to gastrointestinal disease. They might be related to conditions in the hair and in the skin and things like that. Other areas where the immune system is definitely playing a role. So yeah, so autoimmune is one of the sort of key target areas that a lot of folks are interested in testing fecal transplants in and trying to find therapeutics in.
I guess
I don't know of any current trials, but yeah, we're always happy to talk to folks.
The point is sometimes if you have a severe illness and you have a severe disruption of the microbes, the feeling is that just by eating a bit more fiber or eating it isn't enough, that there isn't enough there to actually regenerate. So it works, maybe work better in milder cases, but when you have a really severe autoimmune disease or something else severe, just changes in diet alone may not be enough. You need a kickstart as well.
Yeah.
Next question right here.
You mentioned the sphere of influence of the microbiome. I'm curious about the different kinds of impacts that the microbiome has in the human body. What's being studied now? Does it affect our mood? Does it affect our brain development? Where are scientists really going in terms of how microbiome affects us?
So, yeah, no, it's interesting that you bring up mood. There have been a lot of papers establishing sort of a gut-brain axis, right? And we know that there's microbes that are releasing chemicals that are neurotransmitters. And we know that these neurotransmitters can have an effect on mood, and we know the enteric nervous system is one of the highest concentrations of neurons in the body, and so there's a lot of mechanistic evidence, and there's also some compelling animal experiments sort of showing the power of this gut-brain axis. But it's also one of the kind of big unknowns. So there's maybe fewer people studying that, but in terms of clinical impact, it's really
There's also
huge,
worry,
right?
isn't there, Eric, that these animal studies are showing that you can, if you make some mice anxious and you take their poo and you transplant it to another mouse, you can make that mouse anxious as well. Which, it sounds a bit fun, but then you think about fecal transplants in humans. What happens if the donor was a bit stressed out, and you cure your disease, but you end up with this long-term, these neurochemicals that are being produced that you weren't ready for. So this is
and so in turn you make the FDA anxious.
Yeah.
And then that makes all of us researchers anxious.
Yeah, exactly.
And the cycle is sort of positive feedback. But I think the payoff is huge, and it's one of the things I'd like to see a lot more people work on, because psychiatric disease in general is one of those areas where we don't have the same array of really good molecular diagnostics and really good small molecule therapeutics. So I think it's understudied, very exciting, and potentially a big impact.
Next question here.
Yes, I'm Mike. When a person has a complete clean out prior to an operation, how does the microbiome regenerate on its own, and will it resemble what it was before?
Well, I looked at that. I mean, I asked some of these gastroenterologists doing these colonoscopies, and I also tested my own gut before and after. And generally, it does return fairly quickly to where it was. And we think that's because they know where to hide. Our guts are very long, and there's lots of crevices, little cracks to hide in, and they're very skillful, and they know there's a big tempest comes, they all batten down the hatches, and they wait for the storm to pass, you know, and you've been to the toilet 20 times, and then they come out again, and they say, hey, it's all sunny now, and food starts to come again, and they grow. And that happens in most cases, but most gastroenterologists report that about 1 in 6 people who have a colonoscopy end up with some change in symptoms and can be improved just by that process. So we respond differently. So it could be that in some people, just that process of being washed out can get rid of some of these more toxic bacteria. But in most people, it doesn't. But it's again, one of these real mysteries, you know, of why we react differently and why some people take a long time to recover, some people instantly recover. A bit like after antibiotics. Huge variation between people in how our microbes respond.
This will be our last question.
I wanted to thank the speakers, and I wanted to continue sort of the dialogue, and the great interest has been shown here, and ask Eric, you're setting up a brand new microbe center at MIT. How do we follow the progress of that and learn what interests we have initiated here? How do we follow you?
Absolutely. So you can follow us on Facebook and Twitter, of course. And you can also go to microbiome.mit.edu, where you can get all of the news and events. And we have a blog where we regularly find microbiome science that we think is going to have a short-term clinical impact and really affect patient care. And then we write a blog entry on that and sort of convey that science to the general public there. So the best way is to go to microbiome.mit.edu and find out all about our different events and activities.
Excellent. We want to thank both speakers for their attention and what they've brought to us here. And we're saving some time so Tim can do some book signing of his new book, The Diet Myth, which is really new. Isn't it out just in the last month, Tim?
Yep. That's right.
So thank you very much.
All right, well, thank you very much for joining us tonight for a fascinating examination of the microbes in diet and the thought of stool as medicine. As an insurance policy, remember to feed your body fiber, inulins, and polyphenols, and don't be afraid of dark chocolate, red wine, and olive oil, the good stuff, the fresh stuff. And the good news is you'll never dine alone, and you will have your own little English garden. So thank you very much for joining us. We hope to see you back next week when we have a screening of Mass Extinction. Thank you. Have a great night.